Here we are, AP Biology, Chapter 13. We're talking about evolution or microevolution. This is part 2 video. Part 1 was on Darwin's kind of study and overall idea of natural selection. This video is going to focus on what is the evidence that we have for evolution. One of the biggest pieces of evidence that we have for evolution is fossils. Fossils strongly support the theory of evolution because we can actually document the change that has occurred over time by looking at the remains of organisms that have lived in the past. And we can see very slow and surely what those changes have been. So fossils have provided a huge amount of evidence for the theory of evolution. The fossil record does show that organisms have evolved in a historical sequence. And what does that mean is that they have changed over time. Now, one of the neat things that happens with fossils is that they can link extinct species, so species that are no longer around today, with species that are currently here that we can see. And how do we do this? By looking at the bones. We can tell that organisms that have, for example, here we look, this one is showing leg bones in this video, this little red right here. This is the leg bones. And if we were to look at those bones, we'll see that they're the exact same leg bones, but now they're hidden inside of a fin. A mass of other evidence kind of reinforces the idea. So if fossils alone are not enough to say evolution is happening, we do have other types of evidence. And we have something called biogeography. Now, biogeography actually started back with Darwin when he was documenting all the countries and all the things that he saw. And basically what it shows is that the geographic distribution, so where on the planet animals are found, can actually show us that they've come from a common ancestor. Because of the way that the earth changes, we can actually see. And what was interesting is that the Galapagos animals actually resembled species of South America, those on the mainland, more closely than those that were on similar but distant islands. So the same environment did not select for the exact same features. And the reason for that is that we now know the Galapagos Island broke off of the mainland and drifted away over time. And so those animals used to live together, and because the island of Galapagos drifted away, the climate and the nature selected for different traits. The other type of evidence we have besides biogeography is comparative anatomy. So simply by comparing the anatomy or body structures of different organisms, we can tell if they're related. And then we have homology. And homology basically says, look, if you have the same traits, it means you have the same DNA, which means you have a common ancestor that passed on that DNA. So let's look at homologous structures. These are structures that have completely different functions, so they're not used the same way, but genetically and structurally, they're the exact same. So for example, if you look at the hand of a human, you wouldn't necessarily think that it's directly related to the paw of a cat. But guess what? Bone for bone, we are the same. Just like in a whale or a bat. A bat wing and a whale fin look nothing like a human hand, but the bones inside of those, they're very similar. And the bones are developed from the DNA, which means we have a common ancestor with cats, whales, and bats. The other one is comparative embryology. By simply looking at the embryos as various stages as they develop, we can actually see that we're related to other organisms. And here's some kind of good examples. Humans and chickens both have pharyngeal pouches, so pharynx. And we both have this postanal tail, which is your tail bone. We used to have tails. And our tail has gotten smaller over time, but how do we know we used to have a tail? Because we develop a tail bone. We also had gill slits. Why would we have gill slits? Because we used to be related to ancestors that had gills or could breathe underwater. Now, the biggest one that's kind of happened lately and that is getting the most attention now is the molecular biology. By simply comparing the DNA, because we have that technology now for genomics, or the amino acid sequences, we can actually see how closely related we are. This will be the focus of the next AP activities that we're going to do, looking at evolution. So if we were to compare DNA, we are 98% similar to monkeys or chimpanzees. That's a huge amount of similarity, but we're not even that distant from, let's say, a chicken. If you look at the actual percentages, we share roughly 60% of our DNA with a chicken. Now, what happens is that this is looking at development over time. Well, with the development of research and things, what we have actually found is that we can see evolution happening in real time within generations. And some of the best examples is camouflage that happen when we take an animal and put it into two different environments. So these are very difficult to see. But this is actually a mantid, so a praying mantis. And this is a praying mantis in Malaysia. It has camouflaged itself to look exactly like a flower that grows there. Whereas a mantis that is in Costa Rica has camouflaged itself to look exactly like the leaves that are found in the rainforest of that region. This is the same praying mantis, two different environments. It looks drastically different because of the way nature has selected for traits that allow it to be hidden. Another example is pesticide resistance. Again, this happens quite often. So we have our organisms, we spray them with pesticide. Maybe one of them has a mutation and survives. That one reproduces, produces a whole population. We go to spray with pesticides again, and guess what? They're now resistant. Now, the reason why this matters is because we're looking at populations. We're looking at the big scale and then the micro scale. And so with that in mind, what we need to talk about is what is a population. A population is a group of individuals of the same species living in the same place at the exact same time. And a species is simply individuals that look alike, can interbreed with each other, and produce fertile offspring that can then interbreed and reproduce with each other. Now, the reason why we want to talk about populations is because they're how we can study what changes happen over time. By looking at their genetics, we can connect to what Darwin was saying is his theory. And he was, again, looking at populations of animals at different locations, and based on that, he came up with the theory of evolution. One of the vocab words that we're going to be talking about is called a gene pool. A gene pool is simply the collection of genes in a population at any one time. So the gene pool is constantly changing. And if the gene pool changes, then we know evolution is happening. With that idea, we have microevolution. Micro. What micro means is small evolution. So we're looking at small evolutions that happen. So the evolution within a population. Anytime we change how often an allele or a version of a gene shows up in the gene pool, then we know evolution is happening because we're changing how often a trait will show up. Now, with this idea of microevolution, we have to step back and look at what are some of the non-evolving populations that can happen. So if there's a population that's not changing or not evolving, what has to happen is that the alleles that get shuffled with sexual reproduction cannot lead to a change in genetic makeup, which means that one trait cannot become more popular than any other trait over time. That's a non-evolving population. How do we know if a population is changing over time? One, we can look at it, or we can do some mathematics. And it turns out that the mathematics would really help us show evolution because they can give us a number of how much things are changing. And the math that we do is called the Hardy-Weinberg equilibrium. And the Hardy-Weinberg equilibrium is going to show us how genes that get changed over time alter the amount of frequency it is in the gene pool. And so with the Hardy-Weinberg equilibrium, we have to learn how to do some basic math. Now, Hardy-Weinberg equilibrium exists kind of on the idea of probabilities, like when we do Punnett squares. And so what the Hardy-Weinberg is doing is it's following the alleles in a population and tracking them generation after generation after generation mathematically. Now, in order for a population to be in Hardy-Weinberg equilibrium, which means not evolving, one, the population has to be huge. Two, it has to be completely isolated, which means no migration in or out. There can be no mutations that happen in the gene pool. Mating has to be completely random and no sexual selection can be used. And all individuals have to be equally successful in reproducing. All of these conditions have to happen. Otherwise, evolution will occur. Now, The reason why we use Hardy-Weinberg and why we're going to study it as we go forward in our AP activities is because it can actually tell us the frequency of disease-causing alleles in a human population. So when a study goes out that says, oh, cancer's on the rise, how do they know that? Well, they're looking for it. Or maybe there's a huge increase in a disease for diabetes, and we're watching that gene become more and more common in populations over time. Now, what I'm going to do is stop the video here and change over and then talk about what are some of the things that can contribute to evolution besides or what can change the frequencies in the Hardy-Weinberg equilibrium.